This is a production of Cornell University.
All right, great. Well, wow, thank you very much, Michelle. It's wonderful. I had to pay Michelle to say all that stuff. And even more importantly, I had to pay her even more to not say stuff. And Bill, all I have to say to you is WTF. And I better explain that because there's some looks in the audience like, you can't say that to him. But here's a story about Bill. Several years ago, I was the director of graduate studies when Bill was our department chair. And we had a colleague of ours that was supposed to do something. And then at the last minute, they decided they wouldn't do it. So Bill sent me an email explaining this. And I got rather pissed off. And I fired off an email back to Bill that was basically 3 big block letters, WTF, with an exclamation point. A few minutes later, this bewildered Bill shows up at my door, looking at me going, and I was like, wow, Bill, you don't really understand what that means, huh? And I knew, you know, Bill being a rather, I guess Bill led kind of a secluded life, being from Nebraska. I think the worst thing I ever heard Bill say was, oh, prune juice. And so I felt the need to clue Bill in as to what WTF meant. And so I said, Bill, WTF. Wow, that's funny. So it's okay to say WTF to Bill. All right, can everybody hear me? I'm not sure if am I supposed to wear this thing? Okay. That's why I wore this tie. I could clip on the microphone. It's my virus tie. They don't make plant virus ties. This is West Nile virus. Still a good virus though. So what I wanted to talk to you today about is getting plant viruses to where they need to go. And that's not only how plant viruses move in the plant, but also how they move from plant to plant via their insect vector. And not only that, but making sure that the virus in the plant is truly available to the insect vector. Because if it's not going to go anywhere, right? So Bill already kind of did this, and so I'm not going to go through my history before. But what I did want to say to all the students and the younger people in the room is I did a lot of different things, and I just want to say, don't be afraid to go out there and try different things, okay? Don't get so locked into one little aspect of your research that you don't get to experience other things in life. I think it's really important to do that. Me. So one, I have to thank my wife. She is here today. She came to my PhD exit seminar, and she's coming to my Cornell exit seminar. She hasn't been to anything in between. I always dress like this at seminars. This is all she's ever seen me. Okay, and I really thank. She is my soulmate. She is my best friend. I would not have been here without her. I also want to thank these 3 individuals. Dawn Smith. Dawn is not here. Jason is here, and Mary Kreitner is here. These were the lab managers, and Mary was a grant manager. They were my left and right hands, arms, and everything else. They really made things work. I had 3 projects, basically, over my career, one being virus epidemiology and management, the other vector biology, which I'm going to talk about today, and the other is potato virology. And if you really want to learn about potato virology, I have to give another seminar in 2 weeks at Geneva. You're welcome to tune in for that. And there's lots of people that are involved in this, okay? I don't do the stuff, you know, all these nice things that people say about me, it's really all them. And you might wonder why I have a picture of this guy, and some of the younger people probably don't even know who this is. This is Ronald Reagan. He was the 40th president of the United States. Why do I have Ronald up there? Ronald is the reason that I'm here today, to tell you the truth. Ronald got elected in 1980 when I was working at the environmental consulting firm the first thing that Ronald Reagan did was roll back all the environmental regulations. Sound familiar? Happening again, okay? I saw the writing on the wall and said, I better get the heck out of this profession. I was plenty happy riding around in my research boat, you know, eating lobster and going fishing. It was a lot of fun. But when Ronald came in, the environmental consulting business went down the tubes. I went back to graduate school. There's one other thing that I learned from Ronald Reagan. Ronald Reagan was a B-grade actor who became a politician. I think Ronald knew that he was not the brightest bulb on the tree, but he did one thing, and that was he surrounded himself with really smart people who made him look really good. And that's been the way I operate as well. Surround yourself with all these smart people, they will make you look good, okay? All right. All right. So enough of that nonsense. Let's talk about luteoviruses and how they move. And luteoviruses do cause some really important diseases. They cause yellow dwarf disease in cereal crops. They cause this net necrosis in potatoes, and they cause a lot of other diseases. They are transmitted by my favorite thing in the world, aphids. Not favorite person. I'm going to show you a little movie here, which I did not make this movie, but I think it's great. This sort of, we understood luteoviruses to be circulative non-propagative viruses. They are acquired from the phloem, so the aphid sticks its stylet down into the phloem where those viruses are floating around, and it starts sucking up virus particles. These virus particles get into the digestive system of the aphid. They move through the digestive system. This all takes about 8 to 24 hours to occur. They go through the digestive system out into the hemocoel where the blood of the insect is circulating. Insects don't have veins and arteries. They just have sort of an open circulatory system. The virus that gets out into the blood ultimately makes its way back up to the head of the aphid, where it associates with the accessory salivary gland, which is actually just 4 single cells. And it goes back through the accessory salivary gland, and then when the insect starts feeding on another plant, it spits out things, including the virus, and that's how viruses get transmitted. These viruses are retained in the aphid. As soon as the aphid sucks it up, it's retained for weeks, or essentially the life of the aphid. The virus is not lost when the insect molts. The other thing is not all ingested viruses will circulate through the aphid, and that's something to think about. Now, when I started here, Bill Rocco was my predecessor. Bill left here in 1986. And Bill left me this experimental system, which was really cool. Bill spent 30 years developing this, and I was really fortunate to have him hand this over to me. This basically was 5 different viruses, all related, all sort of, they're now species, but they used to be just strains of this virus, the yellow dwarf virus, with these different aphid species. And you can see this is transmission efficiency. So Cetobium aveni is really efficient at transmitting these 2 viruses and not so much this. And so this whole system, and Dawn Smith was a technician with Bill, and then she was with me for 25 years or so. Dawn was really the one to develop this whole, this is about 60 years worth of data. I think this is probably the longest, biggest data set ever. But you can see that it's really interesting that some aphids transmit some viruses and not others. These are all related viruses, so why is this happening? And that was sort of the basis of how I would spend the next 30-something years, right? Trying to figure this out. Now, aphids, if you're not familiar with aphids are really cool. It's a female society, right? This is a female, and it gives birth to live young. And it will continue to do so for her whole life, and she'll give birth to lots and lots of live young, which will then grow up and give birth to lots and lots of live young. And these are clonal, right? We've maintained these aphid species here for almost 60 years, and they just clone themselves. And you get lots and lots of aphids, so tons and tons of experimental material. They're genetically identical. It's really a cool system. Couldn't have done much without it. Now, Fred Gildow was a Penn State professor. He is my good friend and was a wonderful colleague and cohort through all of my career. Fred was the one who sort of figured out, while he was a student with Bill Rocco, so in the lab that I have now, Fred was the one who figured out how luteoviruses move through the aphid. And this is just a cartoon of, so this is the stomach of the aphid here, the lumen of the stomach, and how viruses move through the epithelial cells of the midgut into the hemocoel, or the blood of the insect. And it's not just a simple just uptake any virus and it moves through. It's very specific how these things bind to receptors, move through in vesicles, and come out the other end. And so viruses can be blocked. If they can't associate with this membrane, they won't move through. That's one way that viruses don't move through the aphids. This is at the accessory salivary gland coming through. So it's in the blood and it wants to go out into the salivary duct so it can get spit out into plants. And again, it's very specific. It can get blocked by this basal lamina, or it can get blocked at several different steps as it's heading out into the salivary duct, okay? So we knew this kind of stuff. What we didn't really understand was how this all worked, all right? And this is where we started looking at what's the aphid's contribution to this whole thing, okay? And Mary, you met on the video, and Cecilia, you met. All these three were really key players in this, looking at understanding the genetics and proteomics of aphids and aphid virus interactions. The other nice thing about aphids is they come apart really easily. Great, okay? You can rip the heads off aphids. Here's the salivary glands right up here. Nice, accessible. Here's the whole stomach, midgut, hindgut, stomach area. Okay, all the parts that are good for virus transmission come apart really easily. So it's kind of nice. All right, so one of the things that Mary did, and this is Mary here. Can you see that? No, that's not showing up. Can you guys see this arrow from Geneva?
Dominic is coming.
Okay.
No, you can't see that. But
all right, never mind. Sorry about all this. And this is Marina Caillot, who is in the Department of Entomology here. Marina is an excellent aphid geneticist. What Mary did was take one species of aphid that we used to use and two species of the virus. And she had different biotypes of this aphid. So they're all the same species, they're just different biotypes, and here's a bunch of different biotypes. So they can look different, but they're the same exact aphid. But you can see here that these biotypes over here transmit the viruses, whereas these ones over here do not. Okay, and as I said, aphids are just basically female society. It's females who don't really need males, but if you change the environmental conditions, you can get aphids to produce males and females, sexual males and females. Okay, and that's what Mary did. This whole slide and what I'm going to tell you is like 4 years worth of work, okay? But what Mary did was to take this one here, which we could get males from, and she mated it with this one from South Carolina, which doesn't transmit virus. She did an F1 and an F2 population, and she basically got 92 colonies of aphids. These are all clonal colonies, okay? So this is the population. And she did transmission studies to find out whether there was any sort of genetic segregation in this population, okay? To find out whether they could transmit these two viruses, and also if they couldn't transmit the viruses, whether it was a block at the gut or a block at the salivary gland, okay? And so I'm just going to give you the Cliff Note version here, is that basically transmission is a heritable trait. It's segregated for virus species transmission, okay? So whether it transmitted these two viruses, there were some clones that did, some that didn't. They also segregated for the barrier. So some were blocked at the hindgut, some were blocked at the accessory salivary gland. Transmission through all the analysis that I can't go into, but it was regulated by multiple genes that were additive in effect. So aphid genes, multiple aphid genes, additive in effect, and the transmission of these two viruses was controlled by one major gene that controlled both viruses, as well as a bunch of minor genes which were additive, which were virus specific, okay? So that was some really interesting stuff. The next questions that she was asking is, well, why can a non-vector genotype, why is it unable to transmit virus, okay? They ingested similar amounts of virus. They retained similar amounts of virus over time. It didn't go away. And also the virus did not degrade in the hemolymph or didn't change in the hemolymph, okay, to become non-transmissible virus. And the way is a kind of a neat experiment. The way that you do this is you feed the aphids on your virus, and then you bleed those aphids, get some of its blood out, and you inject that blood into another aphid, which is really good at transmitting that virus, and you ask the question, is that virus still transmissible? Okay, and that's the way you figure out, is the virus degraded in the hemolymph, or has it changed? And it didn't, okay? So nothing is different between a vector and a non-vector. So what's the deal? So what's the difference? Is there differences in the aphid virus interactome? Okay, well, enter Michelle, okay? And Ted Tannhauser, who's a proteomics guy out at the USDA lab. And Michelle started, she wanted to get into proteomics big time, and she has, obviously, but she started doing this 2D differential gel electrophoresis, which is essentially take all the proteins in the aphids, the vector and the non-vector, run them out on a two-dimensional gel, and ask the questions, how many proteins are different in the vector and how many proteins are different in the non-vector, okay? And so all the yellow spots is the proteins which are common to both, and the green ones are just found in the vector and the red ones in the non-vector, okay? So you get, you know, this nice, beautiful Christmas tree effect here, but the question is, you know, how many of these are really involved in transmission, right? I mean, there's hundreds of differences, and each of these spots is not necessarily just one protein, it could be many proteins, right? So what Michelle did was pairwise analysis of all the parents, both the parents and multiple ones of the F2s that we knew were not vectors or were vectors, and whether they, if they weren't vectors, whether it was a block at the gut or the accessory salivary gland, okay? And again, the Cliff Note version of the results is that not only did she find aphid proteins that were linked to transmission, but she also found Buchnera. And Buchnera is an endosymbiont. It's a bacterial endosymbiont that lives in the aphid and supplies the aphids with essential amino acids that the aphid doesn't make, as well as do other things, okay? So this was really cool that we found these bacterial proteins, which were also involved in transmission competence. And of course, these proteins lined up with a lot of different things. That's not saying that that's what they did. They just, that's what they were in the database, okay? Proteins were also linked to specific transmission barriers. Like there was 8 of them were related to gut transmission of the virus. There was 5 of them related to the salivary gland. And really interesting is that many of these proteins were isoform pairs. So it wasn't that the protein is missing from a non-vector or missing from a vector, it's that the proteins were slightly different. And had Michelle done anything else beyond this 2D dyes gel analysis, she would have never identified these isoforms because the protein is present in both vectors and non-vectors. So this was like a really cool, really big step. She got on a couple of covers. Thank goodness for Kent Luffler. I really miss him. He was our photographer here for a long time, but this is really, and there's a lot more to this story, but I just don't have time to get into it. Ask Michelle. At the same time that people were working on the sort of the aphid side of things, we were also working on the virus side of things. Okay, and what does the virus contribute to this whole transmission process, right? How does that work? Now, we did most of the virus work with potato leaf roll virus, not the cereal dwarf viruses, because we didn't have infectious clones at the time of the cereal dwarf viruses. But these viruses are very similar. This is the genome of potato leaf roll virus. It looks sort of like RPV. The thing I want to point out is there's 2 subgenomic RNAs that are used to translate. This subgenomic RNA 1 is my important one here, and it translates the coat proteins and the structural proteins as well as a movement protein. Okay, and that's what I'm going to focus on here. So, looking at this piece of the genome, I'm going to focus on these 2 proteins. This is the coat protein, and this is the read-through. The read-through protein is this whole protein, right? It gets read through here at this stop codon. This is the read-through domain. The coat protein, this virus is an icosahedral virus, okay? And so the coat protein makes up a majority of the virus. And then this read-through domain kind of sticks out on the surface. Now, this is not exactly what a ludiovirus looks like because Michelle hasn't finished her work and she's got to tell us what this actually looks like, but this is pretty close, okay? The read-through domain does stick out on the surface of the virus, okay? But this protein here, this read-through protein, is also found as a soluble form roaming around the plant, not associated with virus particles. And so it's interesting, in a virus particle, it's actually this read-through domain is actually cut off. It's only this piece in the coat protein, which is in the virion. This piece here is missing. But this whole thing, this thing here, wanders around in the plant. Okay, so there's virions, and then there's this soluble protein. Now here's about 20 years worth of work on this slide, okay, and I'm not kidding, and there's a lot of people involved in this. Kathy Che was my second postdoc, and she's really the one who got us involved. Kathy's now a senior scientist at Monsanto and has been for a number of years. And some of you may recognize Peter Poliakaitis, who was a professor here before moving on to Scotland, before moving on to Korea. But Peter's been a long-time collaborator on this. But what we did was, by a whole variety of techniques, a lot of mutational analysis, was figure out what these proteins do, okay? And it's not just what the protein does, it's actual many domains within each piece of this protein that actually works to do something, okay? So like the very beginning of this coat protein is involved in interactions with the RNA inside the virus particle. And the whole thing about this is that disorder, chaos, okay, is the key to this whole functionality of this read-through protein. Okay, so this is a sort of a disordered piece of the protein. This is the ordered sections of the proteins. Those are very stable. Okay, so disorder, you would expect that the protein has to reach inside and play around with the RNA. That might be a disorder, and it indeed is, whereas the virion structure is going to be very tightly conserved, and that's this section down here. These domains down here also contain several domains which are responsible for moving this virus through the gut of the aphid. Okay, and then we go through this very disordered section here, which is actually a proline hinge that allows this read-through domain to be kind of flexible on the surface of the virion. Then we go back into a very ordered section of the read-through domain here, which is involved in a lot of different things, from assembly of this read-through protein into the virus particle itself. It's involved in translation of this read-through protein, and it's involved in how this virus moves through the gut of the aphid. Okay, and then we go back into this really disordered section, which is this terminal end, which is not part of the virus particle, but it is part of this soluble protein. Well, I think one of the more interesting things we found, and this was by Carrie Peters, who was first a graduate student and then a postdoc for a while, was that Carrie was playing around with these proteins, and she noticed, so if you put this whole protein, the virus, into the plant, and you look at it, it causes these kinds of symptoms in about 3 to 4 weeks. And this is a phloem-limited virus, so if you look at where the virus is in the leaf section, it's always associated with phloem bundles. Okay. Well, Carrie made a mutation where the RNA is still there, but it just doesn't translate the whole protein, right? It cuts off this section of the protein. And when you do that, when you inoculate these plants, you have to wait a little bit longer, about 8 to 10 weeks, but the symptoms changed. And when she looked at where the virus was in the plant, the virus was no longer associated with phloem. It had leaked out into mesophyll tissue. Okay, which is really kind of interesting. This is a phloem-limited virus. Phloem-limited viruses at the time were thought to be phloem-limited because they didn't have proteins which allowed them to move out of the phloem. Okay, this suggests something totally different. The other interesting thing is that you can't feed aphids on these leaves and get transmissible virus. And think about this. These aphids feed in the phloem. They pick up virus from this, but they won't pick up virus here because the virus isn't in the phloem. There's a lot more virus, but it's not accessible to the aphids. Therefore, it's not transmissible. Therefore, no problem, right? Well, that was really kind of cool. Now, my lab runs on serendipity and laziness sometimes, okay? And this is where serendipity and laziness comes in the first time. So Carrie had this, right? This virus, which was no longer Phloem limited. And she had this construct, which was completely serendipitous because during the whole cloning process, it wasn't supposed to be like this, but what had happened is that there was a change during the cloning, and we got this weird clone, which had this deletion in it, and it actually changed it had a frame shift in it, so it no longer produced this piece of the protein, okay? So it caused this, right? Which we kind of expected. But Carrie, being lazy, she never threw anything out, right? I guess it was like, well, if I have space in the greenhouse and I keep plants there, then it will always be my space and I don't want to give it up. So I kind of keep my plants, and she kept them around for a couple months, and she noticed that these plants here all of a sudden look like these plants. They kind of went back to the normal. And I was kind of like, well, that doesn't make much sense, right? You must have gotten contamination. Well, she sequenced these viruses, and in fact, this virus is really resilient. It self-imposed a Phloem restriction on itself. It changed. It actually, like, selected for these mutations, whether it be other deletions or indels or other SNPs, which actually changed the reading frame and brought back this piece of the protein. And when it did that, the virus becomes Phloem limited again. So the virus is actually sort of selecting to be Phloem limited, which kind of makes sense when you think about aphid transmission. Now, the other thing, keep talking about serendipity, is that Yi Zhu, who was a postdoc, working kind of with Alan Miller, who's a really good colleague out at Iowa State. Alan had discovered some years ago that the translation of this read-through protein was some way controlled by some element here in the middle of the protein, which sort of went back and connected with something at the beginning of the protein to actually regulate translation. Okay, but the whole mechanisms were not really understood, and so Yi took on this project, and first by doing some mutational analysis, he narrowed it down to this little section here, which is responsible for translation of this protein. And if you look at this section, it's this Stemloop RNA structure, okay? This is kind of neat Stemloop structure, all right? And so if you look at this, so he made some mutations in here, one that would disrupt this stem or this loop or this loop or this loop here, and made some mutations and asked the questions, does that affect translation? And indeed, the mutations up here, if you disrupted this stem or this loop, you did not get translation at 15 days post-inoculation. Yi, being like Carrie, never threw anything out either, okay? And so a month and a half later, he went back out to the greenhouse to look at these plants, and lo and behold, they looked different, right? They sort of looked like wild-type plants again. And when he looked at what was in these plants, first of all, he sequenced stuff, and so here's the original mutant that he made, here's the wild type, and here's the recovery. And so it didn't revert back to the wild type. And you wouldn't really predict, based on the sequences, what was happening. But when you model these sequences, basically, so the wild type has this stem loop structure, the mutant is disrupted in this loop, and the recovery, the ones that are in the plant doing just fine, have sort of pseudo-reverted. They haven't quite reverted to the exact same structure as this here, but this mutant, and there were several others that don't look exactly the same, but in all cases, what happened was that they reverted back so that they could produce this little loop. And when they did that, then translation of the protein was restored. So that was really neat. And Yi has gone on to do a lot of other neat things with this protein, but I don't have time to talk about it because you all want to get out of here. But I'm just going to tell you about one more serendipity thing, okay? And that is, what the heck does the plant contribute to all this stuff? Okay, and this goes back to Carrie, and Michelle was first here, and now Stacy's involved. But back in the day, Carrie was trying to purify virus. Purifying gluteoviruses is a real bear. Okay, you can start with tons and tons of tissue and wind up with little teeny amounts of virus. Okay, and so our transmission our purification protocol is fairly involved. And a lot of times we add these things to buffers to do various things, whether it's to make sure that the virus doesn't aggregate, whether it's to chelate heavy metals or to be an antioxidant. We add these different things. And so Carrie was doing these things. And she noticed that when she just used buffer, that the virus was transmissible. Okay, so she got purified virus, she fed it to aphids, it was transmissible, right? But when she added sodium sulfite, which we add routinely, she had just forgot to add it one time, that the virus was no longer transmissible. Okay, which was kind of weird. Okay, what the heck did you do? All right, and we finally figured out, well, I forgot to add the sodium sulfite. Okay, so that changed something. Okay, now the viruses look the same, but the difference was, and here we're looking at electron micrographs of what's going on in the aphid gut, is that when you just use buffer, the virus moves through the gut into the interior of the gut. Now you can kind of see the little virus particles here. When you use EDTA, which is another additive, the virus works just fine, goes in. But when you add sodium sulfite, the virus doesn't go through the gut. Okay, it's not absorbed into the aphid gut, so it's not transmitted. Okay, so what's going on? Well, come back with Michelle and the great proteomics work that she wanted to do proteomics on everything at that time, right? Well, Michelle looked at this, and just a long story short is that what she found was that there were 20 plant proteins that were associated with the transmissible virus, but were not associated with the virus that wasn't transmissible. Okay. Now, I'm not going to tell you any more about this because Michelle and Stacy and others in her lab have gone on to do all kinds of really cool stuff to figure out all of these plant proteins which are associated with the virus complex and work in the aphids in mysterious ways. But it's not just that virus, and it's not just the aphid, okay? And I'll let Michelle invite Stacy to come and give another seminar, and she can tell you all the great stuff. Oops. So I'm just going to end with, when I first started, the arbovirologists, the people who work on insect transmission of viruses, basically said, look, this is how it works. It's either mechanical, it's a flying needle. Insects suck on sap, get stuck in their mouth parts, they move to a different plant, and they spit it out, no big deal, right? Or is that the insects get infected with this virus and then they infect other things. But that's not how it works at all. Okay, that's certainly not how it works for luteoviruses, because the virus does have to circulate through the insect, but it doesn't infect the insect, right? And mechanical transmission, the flying needle whole thing, that's just complete bunk as well. So I'm just going to end by saying is that virus vector interactions as the basis for transmission is being really naive. This is where we were 30 years ago. It's all these virus vector interactions, when in fact, the virus transmission is much more complex. You have to examine all virus vector endosymbiont and host interactions to fully understand how this whole process is working. Reverse genetics and mutations are great for looking at viruses and what they do. But if you don't move forward and you start looking at structures and you start looking at interactions, you'll never figure it out just based on mutational analysis alone. We need to really look at how the virus manipulates the host and the vector to ensure its survival. And there's a lot of research going on in that area right now. Michelle's involved in that, a lot of other people, but this is turning out to be really cool. We need to understand how these virus-host interactions ensure the virus-vector interactions. The virus has to be in the right place. It can't just be infecting the plant anywhere. It has to be in a place that is available to the aphid or whatever the vector is. And I think that we don't give viruses enough credit. I think that they're far more resourceful than we allow them or want to say they are. I think they're much smarter than many of us, perhaps. So I'm going to end there, but a lot of people ask this question, well, if you're going to retire, what the heck are you going to do now, right? My wife and I, we have a house down in the mountains of North Carolina, up in here. These are the high peaks of the East Coast. Many of you think the Adirondacks are the high peaks. Not even close. Okay, we are going to spend a lot more time with these two little girls. These are our granddaughters. I still have some things going on with the potato industry that I will be doing. This is Grandfather Mountain. Grandfather Mountain is a little under 6,000 feet. This is the Mile High Bridge. And Grandfather, our house is somewhere right down about there. The climate in North Carolina is not much different than it is here. So no, I'm not taking the winters and going to a warmer climate. Just doesn't work that way. Here's Grandfather Mountain in the wintertime. Here's Grandfather Mountain in the fall. It's very beautiful down there. Come and visit if you're ever down that way. I'm still going to be doing my skiing and ski patrol. So Bill, if you ever need a ride down the mountain, I'd be happy to put you in the sled and give you a little ride. Okay, that's it. That's all I got for you. Sorry I went so long. I'm not going to tell you anything that, after what Michelle told you, so don't ask those kind of questions. Like I said, if you want to know about potato viruses, come in 2 weeks. Any questions, Geneva or anywhere else? Oh, Mark.
Yeah. Stewart, did you ever figure out why Pathology is such a good transmitter of BYDV?
And not the other aphids? Well, it depends. I mean, what Mark is asking is one of the species is Rhoploscyphin padi, and it's a good vector of a couple of the BYDVs, but not other ones. And so we have other aphids that transmit, as well as padi, maybe not the same viruses. I wish I could answer that question. That was sort of the question that I was asking at the very beginning. We failed. I failed, okay? I mean, I think we know a lot more about what's going on, but we don't. We know that there are proteins specific for certain species of aphids that interact with different host proteins and different virus proteins, and I suspect that a lot of it has to do with that. But you're going to have to talk to Michelle, because I mean, essentially, I have given everything to Michelle. She's really moving forward with that kind of stuff, as well as a lot of other stuff that she does. But did we figure it out? No, I failed. Sorry, that's why I'm leaving. Anything else? Yes.
So what's up with these reversions of the mutants that are unable to be contained in Phloem and move to mesophyll, and then they revert to wild?
Right. Well, we made the mutants and made them move out, right?
They revert.
Right.
What is the selective pressure for this?
Well, that's a good question, because the selective pressure is in the absence of aphid transmission. So it's not just that there's, that they're reverting so they can be transmitted. They're reverting for something else, and they're reverting to be back in the phloem. Why do they do that? I don't know. I mean, they seem to replicate just fine in mesophyll cells, right? I mean, you can do it in protoplasts and everything else. So there doesn't seem to be a reason why they don't like mesophyll cells. But for some reason, they select for those pseudo-revertants. And if you, going back to the literature, there's a lot of pseudo-reversions that people have found in terms of aphid transmission, not necessarily looking at other factors. But, so pseudo-reversion is like a real, comes up again and again and again with these viruses. But I don't know what the selection pressure is. It's a great question. Michelle, figure that out.
So Stacy does have some sort of, recent work on that we've published and that's coming out. But there's a big, defense response in mesophyll with signaling from the chloroplast. And so, one possibility is that in the phloem, they're sort of buffered from cross pathways and that sort of thing.
See, I told you she.
A follow-up on that. Do you have any sense of what the mutation rate is in the viruses?
That's a great question. So the question is, what's the mutation rate in these viruses? Well, they're RNA viruses, so theoretically, it should be about 1 mutation for every replication cycle. But I don't know. I mean, I'm not so sure that I believe in that anymore. And I say that only because of our work on PVY, where we have done a lot of that kind of work. And we don't see the level of genetic diversity within a population that we would expect to see. So I don't know. I mean, there is quite a bit of literature now in ludioviruses where there are a lot of recombinants occurring. But I don't know. Do we have a lot of data on?
I haven't really looked at it.
You really haven't.
I mean, I think that he did an experiment where he did have a mutation, but it was still a wild type phenotype, and that one didn't revert. Like, you didn't see any changes in that.
Yeah.
But his mutants, where something happened, like there was low titer, or they moved out, then you get the revert. So it's not like it's just making random mutations over time. It's like there has to be some kind of selection. Yeah.
Yeah. That's another reason why I'm leaving, because I can't remember all that stuff. Yeah, Adam.
So in the experiment when somebody left the sodium sulfide out of the buffer.
Yeah.
So there's evidence that plant proteins associated with the virus are important for uptake by the aphid. Is there any evidence for aphid proteins associating with the virus on the way out being important for establishing infection in plants?
Wow, that's a great question.
I don't think
We've not looked at that, because that'd be tough to try and figure out. I mean, there's so little virus transferred to a plant. It'd be really, really hard to do that. Now, you know, it's interesting because there is quite a bit of work going on, especially in the animal viruses, where the vector is creating an infection site for the virus in the host. You know, and that's sort of injecting a lot of vector proteins into that site where it also dumps the virus, and it helps the virus then create an infection site. We haven't been able to do that with these viruses, you know, because we can't get enough material. But it's really interesting, and I suspect that there is something there. Yeah, let me get this.
So forgive me if I get part of this wrong, but I believe you said there was a part of a protein that had a stem loop structure that was important for transcription or an earlier part of the protein. And then she said that when you mutated the stem loop and knocked it out, it reverted back to a similar kind of stem loop structure.
Right. So
do you have any inferences at all for like the mechanism, or like why the RNA backbone stem loop structures are important for its ability to perform its transcription function?
Well, it's translation. Okay, it's translation. I'm sorry if I said transcription, but yeah, we don't really know. Stem loop structures in viruses are a fairly common structure that's involved in function, but why that is, we know that structure loops back. We've done the work to show where it loops back and binds just prior to the start of that ORF. So it is sort of a binding thing, but why that particular stem loop structure is so important, we don't know, other than it is. We have lots of mutants that, you know, that kind of disrupt that one, and it always comes back to building some sort of stem loop structure. Not always the same one, but similar enough. So it's certainly not just a, you know, linear sort of binding, you know, linear sequence binding to another linear sequence. It has something to do with that stem loop structure in RNA, but we don't understand why.
Have you
Michelle.
considered the possibility that it could change interactions with the ribosomes?
Could, yeah. It might be, you know, because a lot of, yeah, you're looking at, you know, like tRNAs and things like that. Those are all very similar structures like that, kind of stem loopy structures. So yeah, it could very well be something like that.
One last question. More general question. What advantage is it to the vector to be carrying viruses around?
Wow. Well, we used to think none, but I don't think that that's true anymore. Michelle, you want to
wait for Jenny's thesis defense?
She's
got that all
got a
figured
student.
out.
Jenny is working on that kind of stuff, and there's a lot to it. You know, it's, yeah, we used to think that doesn't make any difference at all, but it does. You know, in terms of And you know, there's all this function of virus sort of manipulating behavior, you know, to make sure that the aphid either stays on the plant or doesn't stay on the plant, depending on what kind of virus it is, whether it wants to be transmitted quickly or takes a long time. So there's all this kind of really neat stuff that's coming in that, yeah, you're going to have to wait for all of Michelle's people to start giving seminars because they got some really cool stuff coming along.
So it sounds like there's lots more to come. Let's thank Stewart, not just for today's seminar, but he's such a great colleague, a great mentor, and a great, so thank you.
This has been a production of Cornell University, on the web at cornell.edu.